And just moving into our next session, for a person living with a rare disease, I think many of us have experienced this. It can take 5 years or longer to receive an accurate diagnosis. A patient-led collaborative research network enables research that can lead to new pathways to diagnosis through the discovery of new genetic mutations. Advancements in innovative multi-center collaborations could also lead to predictive tools to aid clinicians in making a correct diagnosis before genetic testing. At Kabuki Syndrome Foundation, we are constantly investigating ways to reduce the diagnostic odyssey by improving speed, precision, and supporting broad, equitable access. Early diagnosis can drastically change a child's trajectory. If a child is managed with a Kabuki-specific medical plan and a roadmap of therapeutic interventions, the child can grow vastly in his or her capabilities. To address this, we are committed to supporting Dr. Siddharth Banka's efforts in the creation of clinical management guidelines. In this next section, Dr. Banka, will discuss recent and ongoing research around improving diagnosis and management in Kabuki syndrome. Following Dr. Banka, we will be joined by Lucas Lang, the CEO of Probably Genetic. He'll discuss how his company is increasing access to early diagnostics using machine learning, telemedicine, and social media. At this point, I'd like to introduce Dr. Sid Banka from the University of Manchester.
So thank you very much to the organizers for a very kind invitation to this very exciting session and the conference as well. So I've been given the task to talk about advances in diagnosis and management of Kabuki syndrome. So I'm Sid Banka. I'm a professor of genomic medicine and rare diseases at the University of Manchester. I'm also a consultant in clinical genetics at Manchester Center for Genomic Medicine. And we've recently set up a rare conditions center dedicated to improving the clinical research and education aspects about rare diseases, which I will be directing. So these are really very interesting and important times for rare diseases in general. Specifically for Kabuki syndrome, I've been researching this condition for over a decade now. Our first publication from our center on this condition was in 2011. So it's been exactly 10 years since we published our first paper in the field. Since then, we've been working on trying to understand the clinical aspects, improve the diagnostics, and also we've been using a number of different models to understand the underlying mechanisms for Kabuki syndrome. So primarily for Kabuki syndrome, we use to study induced pluripotent stem cells, or iPSCs for short, and differentiate them into brain cells and try and understand the mechanisms, and hopefully that would lead to identifying potential treatments. However, today I am not going to talk about all the lab-based research. I'm really going to focus on the recent advances that have improved diagnosis and improved clinical management of Kabuki syndrome. And as Janet very rightly said, this is absolutely fundamental, because if you don't diagnose patients in time and accurately, it's very unlikely that they're going to benefit from all the research advances that are happening. So I will start with improved diagnosis part first. Now, I wasn't able to attend the sessions yesterday because I was running my Kabuki syndrome clinic at the hospital. So if there is a bit of repetition, please pardon me. But perhaps maybe it would be useful to spend a couple of minutes just understanding the basic genetics and genetic aspects of Kabuki syndrome. So as you can see here, As you would all know, DNA makes genes, and genes code information for how a cell should function. And a lot of that information is translated in form of proteins. So here is a cartoon where a DNA, i.e. a gene, is resulting in production of a protein down here. So forget all the bits inside, rest of the bits inside the cell. All we need to remember is that there are genes which produce proteins. Now, you all would know that there are two main types of Kabuki syndrome, so KMT2D, changes in which cause Kabuki syndrome type 1, and KDM6A gene, changes in which cause Kabuki syndrome type 2. Now, a vast majority of patients with Kabuki syndrome have Kabuki syndrome type 1, which is caused by KMT2D. So I'll first explain very briefly how do genetic changes in KMT2D cause Kabuki syndrome in most patients. So if, let us say that this is KMT2D gene, it will produce a full-length KMT2D protein, which you can see here. However, in about 70% of individuals with Kabuki syndrome, there is a change in one of the copies of KMT2D. So remember, everyone will have in every cell 2 copies of KMT2D. And if you have a change in a single copy of KMT2D, such that it results in a smaller protein, not a full-length protein, so almost there is like a stop signal for that protein somewhere in the gene. So that would mean that this protein is not produced at full length, and in fact, in majority of cases, it will not be produced at all. So that would mean that an individual with a stop-like change in the KMT2D gene would be functioning on 1 rather than 2 copies of KMT2D. And that is what we find in approximately 70% of patients or individuals with Kabuki syndrome. I think in those individuals, to some extent, once you arrive at a clinical diagnosis, proving it with a genetic test is relatively straightforward. And many times, sometimes actually, a clinical diagnosis may not be suspected and a genetic test is done and then the diagnosis is found. So for example, nowadays, some diagnoses are even made before birth on prenatal testing itself. However, analysis of such genetic results are relatively straightforward because you can actually very clearly see that there is a stop signal in the gene. But that also means that the diagnosis can be a bit more challenging in the other remaining 30% individuals. And there are various reasons, and I'm not going to cover every possible reason why a diagnosis can be challenging, but I'll just address a few more common scenarios. So for example, there might be genetic changes in which rather than the stop signal which I was talking about, there might be changes just in the sequence of the protein that is produced from the KMT2D gene. So rather than a big stop signal here, there's just a little small red dot which changes the sequence of the protein. Now, there are certain changes which may not really impact on the function of that protein at all, whereas there are some other changes which will be really, really bad for the function of that protein. So it's almost like a spelling mistake. So there might be some spelling mistakes where if you make it, you can still read what the word says, whereas there might be some spelling mistakes which completely alter the meaning of that word. So such changes are called missense changes. And really, to be able to identify which spelling mistakes or missense changes are disease-causing versus which missense changes or spelling mistakes are just which do not affect the function of that protein can be a bit challenging at times. So one of the questions which for last 10 years since the gene was found for Kabuki syndrome is to try and make it better to understand which missense KMT2D changes can cause Kabuki syndrome. So there are a number of ways in which one can address that question. With Victor Fondes, who was a PhD student working in Manchester with me till last year, we looked at this question very systematically. So we compared in control populations what are the types and their frequencies of missense changes in KMT2D genes. So these are individuals who do not have Kabuki syndrome, and they've just undergone sequencing of their genome or exome or all the genes in them, and their changes in every gene has been catalogued, and therefore changes in the KMT2D gene have also been catalogued. So we collected several thousand variants in KMT2D gene from control population. So that's what we call control population. And we plotted them across the entire protein of the KMT2D. And then we compared that against the missense changes that have been identified in Kabuki syndrome patients. And then you can very clearly see that there are certain regions in the KMT2D protein in which, in the control population without Kabuki syndrome, the frequency of KMT2D changes are relatively low, whereas in individuals with Kabuki syndrome diagnosis, their frequency is much higher. And similarly, so this systematic comparison enables us to say which regions of KMT2D gene might be more susceptible. If there is a change, they might be more susceptible to leading to changes in the function of the KMT2D gene. It can go much more in detail, and if anyone is more interested, I've put the reference here and they can read this paper. But this sort of systematic analysis really helps the labs and the clinicians and doctors and geneticists to be able to understand which changes in KMT2D gene can cause Kabuki syndrome. So that's really a very small, short overview of some of the work which Victor did while he was in Manchester. However, in many cases, even this may not be helpful because you cannot get sufficient data about each and every region and each and every specific change of KMT2D gene. So some new technologies have really helped in improving that. Even if you don't know what exactly the change is or what it might be doing, you might be able to do some further lab tests to be able to classify the impact of the genetic change in an individual. So I'm sure in all the scientific presentations that have already taken place in this conference, you would know that KMT2D is important for adding a specific chain called a methyl group or methylation change to histone proteins. So these yellow dots here, dots and stars and circles, are histone modifications which the KMT2D gene is adding. Similar modifications also can happen on the DNA. So here the DNA is wrapped around the histones. So together they're called nucleosomes. So when DNA is wrapped around nucleosomes, wrapped around histones, I'm sorry, these DNA, these histone modifications can occur with KMT2D, for example. And similarly, there are other enzymes which can alter DNA methylation state as well. And measuring histone modification methylation in a lab is quite laborious, it takes time, and it is difficult, and it is expensive as well. Whereas measuring DNA methylation is easier because you can just simply take anyone's DNA sample and you can measure their DNA methylation level. And by measurement, it's not just about the overall quantity of the methylation. You can, to a large extent, specify which regions of the DNA are methylated in an individual. So as I said, KMT2D and KDM6A are both involved in histone methylation. Alterations in histone methylation result in changes in DNA methylation. However, measuring DNA methylation is easier than measuring histone methylation. So if we know all of that background information, we can, there's been a lot of work which has been done over the last 4 or 5 years in trying to study the DNA methylation patterns in patients with Kabuki syndrome. And this is a screenshot from a paper from R.F. Gashi et al. Published last year in American Journal of Genetics, where they showed that you can actually identify a DNA methylation signature for Kabuki syndrome. So that works both for Kabuki syndrome type 1 and for Kabuki syndrome type 2. So this DNA methylation signature for Kabuki syndrome is now known and available to labs. This was a paper from Butcher et al. Again published in American Journal of Genetics in 2017. And it may look like slightly complicated chart, but it's actually relatively straightforward to understand. So what they did was they collected DNA from patients with Kabuki syndrome with proven diagnosis. They had DNA from some individuals who had these KMT2D missense changes about which the significance was not known. And then they also had some samples with patients with another condition called CHARD syndrome, which some of you may have heard of, may be familiar with. And there is some overlap between CHARD syndrome and Kabuki syndrome, such that sometimes patients can be confused with each other. So they collected their DNA samples and they they studied their DNA methylation patterns. And then they used a lot of complicated statistical analyses to really try and classify these DNA methylation signatures. So to make it a bit more clear, I've added anything which sort of concentrates here would match with patients with CHARD syndrome. Any sample that clusters here will match with Kabuki syndrome, and these are control samples. So these are individuals with DNA of individuals who have neither Kabuki nor CHARD syndrome. So these samples clustered very nicely, but if you notice these empty blue triangles, so these are from individuals with KMT2D variants of uncertain significance as shown in the legend in the chart here. And so this person's, initially we didn't know whether this variant was Kabuki syndrome causing or not. However, when you do this particular test, you see very clearly that this sample clusters with Kabuki syndrome, meaning that that variant can be reclassified, that genetic change can be reclassified as a cause for Kabuki syndrome. On the other hand, you see another blue triangle here, which clusters very much with the control corner. So that would suggest that this particular KMT2D change is not causing Kabuki syndrome. It's just a random genetic change, and therefore that patient does not have Kabuki syndrome, and actually they can then be investigated more thoroughly, more deeply, to identify the real cause, the real diagnosis for them. So I think as it is important to identify patients or individuals with Kabuki syndrome, it's equally important to ensure that individuals who do not have Kabuki syndrome do not get misclassified. So actually this DNA methylation signature technology has been a real boost for improving accuracy of diagnosis, not just for Kabuki syndrome, but for dozens of rare genetic conditions as well. And I'm glad to say that we've recently, in collaboration with our Canadian partners, we've set up this test in the UK, and we're probably the only lab offering such test in the UK, and maybe one of 2 or 3 labs in Europe and rest of the world as well. So just extending that theme about missense changes in KMT2D gene a bit further. Now, you might think, okay, there are 2 possibilities, that either a KMT2D change might cause Kabuki syndrome, or it may cause no problems at all. We recently showed that there are certain changes in KMT2D gene, which may cause a condition which is entirely distinct from Kabuki syndrome. So this work was led by my postdoc research associate, Sarah Cubertino, and my clinical genetics colleague, Verity Harton. And the work was published recently in Genetics in Medicine. I think it's 2021. I probably put 2020 by mistake. However, in this work, we identified a number of families and patients who had missense changes in a very specific part of the KMT2D gene. And we then systematically compared their clinical features with patients with Kabuki syndrome, and we found that they were actually quite different. So the main features that we found in these individuals were external ear differences, hearing loss, and I know patients with Kabuki syndrome also have hearing loss, but in these individuals, the type of hearing loss was very different. For example, as you can see, it wasn't because of repeated ear infections or conductive hearing loss, which is more common in Kabuki syndrome. This was actually because of absence of the ears. There were also some differences with eyes. So ocular refers to the eye globe. There were also differences in the tear duct, which are called lacrimal ducts. Many of these patients had choanal atresia, which is basically the opening of the nose is not open, it is closed, and it's actually a comparatively frequent feature in Chart syndrome, which I mentioned in my previous slide. So that's called choanal atresia. Then branchial arch is this region of the face and neck, and there are some, almost all of these patients had some branchial arch differences as well. Several of these patients at a very young age, they had low thyroid levels, so hypothyroidism. And some of these patients, almost quite different from Kabuki syndrome, they had absent or very small nipples, so that's called athelia. Whereas young babies with Kabuki syndrome often tend to have more prominent breasts, which is opposite to what we saw in these patients. Furthermore, there were some very frequent features which are very frequent in Kabuki syndrome, but were absent from these individuals. So none of these individuals showed any motor delay, learning problems, or problems in their kidneys as well. Then we also, because facial characteristics are an important differentiating part for Kabuki syndrome, we didn't think that any of these patients shared facial characteristics with Kabuki syndrome, but that can be a bit subjective. So we did an objective computer-aided facial comparison study as well to show that these individuals had significantly different facial characteristics when compared with Kabuki syndrome. And then I already mentioned the DNA methylation technology to use. We also used that technology to show that these individuals' DNA methylation samples clustered separately from type 1 Kabuki syndrome, from Chart syndrome, and from control. So we could then say that these specific missense KMT2D genetic changes cause a condition which is distinct from Kabuki syndrome, both at the genetic level, also at the clinical level, also at the facial level, and also at the epigenetic level 2. So all I'm trying to sort of say in all of this is that accurate genetic diagnosis is really important to make sure that the right patients get the right diagnosis, and hopefully without having to spend too much time in the diagnostic odyssey. In the short last segment of my talk, I'll now move on to improved clinical management of Kabuki syndrome. So I'm sure lots has already been talked about in Kabuki syndrome, and a lot of emphasis is usually given on type 1 Kabuki syndrome, but to be able to manage patients appropriately, we need to know what their natural history of the condition is. And actually, there is a huge gap in our understanding of the natural history and clinical features of the X-linked Kabuki syndrome type 2, which is caused by changes in KDM6A gene. So we recently, again, this was work done by Victor Fondes, who you've already seen in one of my previous slides, where we studied a large number of individuals who had changes in KDM6A gene, and we divided these individuals into 4 groups. So because it's an X-linked gene, males and females can be differently affected. So we divided the cohort into males and females, and then every group divided into if the patient and the individual had a stop change in KDM6A or a missense change. So so that means that these are males with stop change, these are females with stop changes, these are males with missense changes, and these are females with missense changes. And when you plot their clinical features, you do see a very obvious trend in that. So I'll just take you very quickly through some of these measurements. So these are birth weight, birth length, and birth head circumference. And these are weight, length, and head circumference at last examination. And this is the center line, 0.0. So that's completely average mean of distribution for a person's age and sex. And as you can see, all individuals, almost all individuals, their birth weight was around about what would be average for a population. The red line here shows that this is +2 standard deviation. So any child is born whose birth weight is beyond this would be large for gestation. Any child whose birth weight plots below -2 would be small for gestation age. Similarly, again, at length, at birth, the lengths of individuals with type 2 Kabuki syndrome were also very much within the normal range. So normal range would be defined between plus and -2 standard deviations. And again, head circumference as well is within the normal range. There are a few outliers, but more or less, this was all within normal range. However, when you follow them up, you start seeing a very clear trend that their weights start to drop, as you can see. So many of these center points are below -2 standard deviation, apart from females with missense changes. So the dark blue is for males with stop changes, light blue is males with missense changes, dark pink is females with stop changes, and light pink is females with missense changes. So you can again see very clear trend that males with stop changes are most severely affected, and then females with missense changes are least severely affected. Similarly, in terms of length as well, there is a trend for length to start dropping as well, but the trend between the groups is consistent. So males are more shorter, and females with missense changes are less shorter. And you also see a very marked difference in head circumferences, so they start falling below -2 standard deviations. And I think all of this also gives us interesting biological clues about potential mechanisms for KDM6A. So it improves our clinical understanding, but also might help in our biological understanding of the condition. And then we also plotted a number of different clinical features and their frequencies across the spectrum and divided them into 4 groups, as I've just mentioned. Again, I won't go through in detail, but if anyone is interested, the publication is again in 2021, Genetics in Medicine by Fondes et al. And I think it is freely accessible to anyone. But overall, the message is that males with stop mutations most severely affected, and females with missense mutations or changes are less severely affected. And this is my second last slide. The last bit is that what we need beyond understanding the natural history of the condition is to understand what is the evidence base, and can we actually use that evidence base systematically to manage individuals with Kabuki syndrome in the best possible manner. So there was a very nice presentation in the previous session talking about accessing and systematically studying the publication, 900 plus publications about Kabuki syndrome. So there's a lot of data which is already out there. So we have just created a group of families, patient support groups, and individual experts, clinical experts and research experts and scientific experts. And as I said, families as well, people who understand the genetics of Kabuki syndrome. So really putting all of the sort of pieces of jigsaw together to really take forward these management guidelines for Kabuki syndrome project. And these experts and individuals, families, everyone, we come from all parts of the world. So this is not just for UK or US or just for a few specific countries. It would have input from all over the world. Within the clinical expertise, we have expertise of almost all relevant specialities for Kabuki syndrome. So in brain, neurology, ophthalmology, eyes, ear, nose, and throat, dentistry, cardiology, respiratory system, the gastrointestinal system, skeleton system, kidneys, and endocrinology, etc. So almost all specialities which are relevant for Kabuki syndrome. And we've been very lucky that individuals have been really generous with their expertise and time in this project. And hopefully all of this would mean improved diagnosis and management. And when therapies start to come on board, they'll also provide benchmarks for actually measuring whether our management of individuals with Kabuki syndrome is getting better or not with those treatments. So I really think that this is a very important project going forward for the Kabuki community. Would really like to thank Kabuki UK for their support in this project. And it's been a bit slow, unfortunately, partly, I mean, we can all, things have been a bit difficult for lots of medical systems recently because of obvious reasons such as COVID, etc. But we're really hoping that we've set the foundation and we can actually motor ahead towards completion of this project next year. So I'd just like to finish this with a quick summary of what we've talked about. So we talked about how identification of regions of KMT2D gene where missense changes are more likely to cause Kabuki syndrome is helping in improved diagnosis. Similarly, DNA methylation signature technology, how that can help to determine the significance of genetic changes in Kabuki syndrome genes, either KMT2D or KDM6A. Then we've also mentioned that there are some specific changes in these in KMT2D gene that may not cause Kabuki syndrome but may cause something else, and therefore just reiterating the importance of accurate diagnosis. You also touched on the importance of clinical delineation and the sex differences and the relationship between the type of genetic changes and clinical features in type 2 X-linked Kabuki syndrome caused by KDM6A. And then I introduced you to the evidence-based management guidelines project for Kabuki syndrome as well. So I'm hoping that next time when we meet, I'll be able to give you, I'll be able to say that these guidelines are there and they're ready to be used by the community. So I think that's the end of my presentation, but I'd really like to thank so many people, which I cannot really list on this slide. There are actually dozens and dozens of colleagues, individuals, but also Kabuki Syndrome Foundation, Kabuki UK, and of course all the families and individuals with Kabuki syndrome without whom none of this would have been possible. So I'm happy to take any questions maybe at the end of the next talk then. Thank you very much.